Okay, you guys, so yesterday at the end of the hour you were trying to fill in the blanks about what we talked about with how a neuron transmits a signal. And so we, you did the resting potential, right? Did we? Okay, all right, this is where we're at then. All right, so the difference in electrical charge between two regions is called the potential difference. I think we did this together. Did we? Just find it in your notes. In a resting neuron, the potential difference between the inside of the cell and the outside of the cell is called the resting potential, which is -70. It's underneath this neuron picture. Membrane. All right, got it. And so the resting potential is -70 millivolts, meaning that, remember, the inside compared to the outside of the cell is negative. So it doesn't mean that it's actually literally negative, it's just less positive than the outside. And so the neuron needs a stimulus. So check your answers here. This could be a change. This could be a change in the external environment, such as a change in temperature, light, or pressure. The stimulus also can be a neurotransmitter from another neuron. So it just depends upon if this is a sensory neuron, the stimulus is going to be one of these things here. If it's a neuron that's receiving another signal from another neuron, that stimulus is the neurotransmitter, which is what we talked about yesterday, the example I gave yesterday. So when a neuron is sufficiently stimulated, the resting state becomes disrupted, and the membrane becomes what we call depolarized. So it becomes more positive, so about +30. So why that happens? The cell membrane suddenly and very briefly becomes more permeable to sodium ions. Sodium gates open, sodium comes in. Sodium ions rush into the cell through open sodium channels, causing the inside to now become positively charged with respect to the outside, which is depolarization. So that's the first step. So on that graph, that's the part that went up. If the stimulus is strong enough, more sodium gates open, increasing the inflow. This is an all or nothing event. So once that threshold is reached, and the inside of the cell depolarizes enough and becomes positive, so the sodium gate here is going to open, inside the cell is going to be positive, and it's going to continue on, and so therefore will occur. So what that means though is, like, for instance, if you got a little bit of sodium, so you have a few neurotransmitters that bind and the cell becomes a little depolarized, that doesn't mean that you have just a small little action potential. If it just becomes a little bit depolarized, nothing will happen. It has to reach the threshold level. It's become positive enough to open those first sodium gates, and once that happens, it continues on. And so somebody in my first hour shared the analogy that their psych teacher told them it's like pulling a trigger on a gun. So, once you put enough pressure on the trigger, it's going to fire no matter what. All right, and so those are all or nothing event with this as well. Once the inside of the cell becomes positive enough, it triggers the first sodium gates to open, and then it's going. All right, nothing is going to stop that. Question?
Okay, so when it becomes more positively charged on the inside of the cell, that's depolarization?
Yes, because polarization means that it has a charge difference. Okay. So when the cell starts out, it's polarized.
Right.
All right, the inside of the cell is more negative or less positive than the outside of the cell. So then as it So it's, as it goes back to kind of like 0, and then it's called depolarized. Okay. Then it's going to become repolarized.
Yeah.
Okay. All right. Yep. And so the neighboring sodium gates are opened, and we get what we call this impulse propagation going from node to node. And that's a chain reaction occurring just at the nodes of Ranvier. So then we have the repolarization. This change in potential difference lasts only for a brief period of time because the cell quickly acts to reestablish the resting potential. So that's the potassium gates, the sodium gates shut and the potassium gates open, and the potassium leaves and bringing the potential back down to negative compared to the outside. That's what that means. So this is important. Rapid repolarization is important because if a certain nerve cell does not repolarize, it can no longer respond to stimulation. So it repolarizes and puts it back to normal. And that moves from node to node until we get to the end. So right after the sodium gates open and the inside becomes positive, the potassium gates open and the sodium gates shut. The potassium leaves the cell and the cell becomes negative with respect to the outside again, and this is repolarization. So you're polarizing it again, bringing it back to where it starts. So this rapid sequence of changes involving depolarization and repolarization is called an action potential. So at this node, it becomes depolarized, the sodium goes in, triggers depolarization here, but then it repolarizes, and that's an action potential. So that's the graph that we looked at yesterday. So then let's talk about this part of the graph called hyperpolarization. Where it says by the time it's about negative 80 millivolts, which is a little bit more negative than the resting potential here. By the time the potassium gates close, there's been more potassium outflow necessary to reestablish the resting potential, so it's kind of slow to shut, and that's what we call hyperpolarized. Let me just show you here. This is this part of the graph here. So it goes, here's that resting potential. And so this hyperpolarization. So now we have when the sodium and potassium, so sodium came in and potassium went out in this, and more potassium went out, which caused it to hyperpolarize or go below negative 70. So then what happens is a couple of things that reestablish the resting potential, because notice it doesn't stay hyperpolarized for long. So the first thing is that hyperpolarization, that negative charge, changes the potassium channels so that the potassium channels, remember when we talked about the resting potential to begin with, the sodium potassium pump, let's just review here, pumps out 3 sodiums and 2 potassiums in. And then potassium goes from high to low concentration, so high is inside the cell. So we have more of those potassium going out, less sodium coming in. So this negative 70 is reached with this negative charge, the negative 80, the dip here, causes a change in these potassium channels. So we have less potassium going out, so we don't have as much going out anymore, but we still have sodium coming in, and we'll eventually reestablish this more positive charge inside of there. And then the sodium potassium pump keeps it at the negative 70 and reestablishes the gradient. So when sodium came in and potassium went out, if we never reestablish and put more sodium back on the outside and more potassium on the inside, we wouldn't have a gradient, and so then when these gates open, there wouldn't be a high concentration of sodium on the outside to come in. So the sodium potassium pump helps with reestablishing so that you always have those gradients, so that you always have sodium being able to go from high to low inside the cell and into the inside of the cell, and potassium high to low to the outside of the cell. So the sodium potassium pump is really important with that. But the other channels, the potassium channels, change during this, and so they stop taking out the potassium, so you have more positives coming in, and so therefore reestablishing the resting potential. And so then it goes back to normal, to its resting potential, and then can receive another stimulus. So while this is more negative 80, the neuron can't fire, and the reason why is because when these neurotransmitters bind, it has to make up for that negative, so it has to even become even that much more positive. You have to have that much more sodium coming in to trigger the action potential, so it doesn't happen until that resting potential goes back to normal.
So when you have that hyperpolarization, does that happen every time after an action potential?
Yes.
Okay.
Yes. But it's only, I'm talking 1 to 2 milliseconds. I mean,
Yeah.
it's like the time period is very, very small. So it doesn't have a huge effect on, overall, the signal
So when
transmission.
you have it do that, it pumps in more, or it keeps more. Yeah.
Well, yes,
Okay.
yes.
Does
All
it
right.
keep it or like put more?
It keeps so it stops so it keeps the potassium
So
from
it stops it
leaving.
from leaving.
So you have less positives leaving and you have more positives entering. So therefore, the charge becomes more positive.
Okay.
Does that make sense?
Yeah.
Okay. All right. And so, so it goes back to normal. And once it goes back to normal at the resting potential, then all of this works as normal. So the potassium still leaks out, and then this helps to keep it at that particular resting potential. All right. So, I skipped it. This one didn't I? So there's a period of time called the refractory period. So the neuron, this is a period of time where the neuron will not respond to a new stimulus during this time. And so during this time, the original distribution of sodium and potassium is reestablished from the methods we're just talking about. Once ions are returned to the resting potential at -70, then the neuron is ready for a new stimulus. And we're going to talk a little bit about the speed of this action potential affected by the diameter. The larger the diameter, the faster the speed of that impulse here. All right. So, so let's look at this conduction of the action potential. At the site where the action potential is generated, an electrical current depolarizes the neighboring region of the axon membrane. So it becomes depolarized, all right, and then the neighboring region, all right, also then will become depolarized. Action potentials travel only in one direction, towards the synaptic terminals. And this third bullet talks about why. Inactivated sodium channels behind the zone of depolarization prevent the action potential from traveling backwards. So let me explain that third bullet here. So when sodium rushes in here and the cell becomes depolarized and becomes positive, it triggers these sodium gates to open, so this part becomes depolarized. And then when this triggers these sodium gates to open. The thing is, let's look at this middle node. When sodium rushes in here and triggers these sodium gates to open, what makes it not trigger these guys to open back over here and the impulse to go that way in the wrong direction? And that's what they're talking about, inactivated sodium channels. So there's a little bit more, more in depth, but there is an inactivation kind of gate that blocks the sodium channel. So while this potassium gate is open, and when I say that the gates are shut, there's also an inactivation gate that what happens is when the sodium comes in here, it opens this gate, but then there's an inactivation gate that when the sodium comes over here, it can't reactivate the sodium channel and cause more sodium to come in this way. Because if you did that, then you'd have an action, the impulse going in this direction and this direction. So they inactivate the gates behind the node that's being depolarized, so that the only one that is depolarized is the one that is closer to the synaptic terminal, and so that's how it moves down there. So there's a little bit more in depth than that, but that just gives you the idea of why it moves towards the synaptic terminal. And so this picture here just shows you the movement or propagation of the signal down the axon. So how this picture relates to this picture. So pictures 1, 2, and 3 are showing as time goes on. So if I relate that to this, like here's your open sodium gate. So here's sodium open. They're not showing you the gates here, but the sodium gates are open and sodium's coming in. This is at one particular node. And so this opening of these sodium gates are going to trigger the opening of the sodium gates at the next node. So it's going to open here. So what they're not showing you is there would be Schwann cells here, all right, with the myelin. So these are the sodium gates at the next node there. While this sodium is coming in and depolarizing the cell at this node, which was this one here, this one now, the sodium gates are shut and the potassium gates are open. So the potassium gates are open and the potassium is leaving. And so that's how that relates to this. And they're showing you the gates on both sides there. So then the next picture is showing you these sodium gates open, and so the cell is becoming depolarized, which triggers the sodium gates at the next node to open. Sodium gates at this node shut, potassium gates open, and so on and so forth. And so therefore it moves down the axon. So this is in time what happens from one point to the next. So a couple of things make this happen very, very quickly. This happens extremely quick. The first thing that is involved is the diameter. So the speed of an action potential increases with the axon's diameter. Invertebrates' axons are insulated by a myelin sheath, which we've talked about, which causes an action potential speed to increase. Myelin sheaths are made by, we've talked about Schwann cells. Schwann cells are wrapped around the axons in the peripheral nervous system. Oligodendrocytes are in the central nervous system. So myelin sheaths are made by oligodendrocytes in the CNS and Schwann cells in the PNS. So there's different types of cells, but they both make myelin. And these action potentials are formed only at these nodes of Rambier, which are those gaps in between the myelin sheath, where the voltage-gated sodium channels are found. So if you recall, the myelin is made by these Schwann cells in the PNS or oligodendrocytes. Myelin, let me show you this picture here just a second, let everybody catch up. So this picture here, I showed you before, but the cross section here, here's the myelin sheath, and this is the Schwann cell. Remember we talked about how the Schwann cell wraps around the axon. And the Schwann cell, though, secretes this molecule called myelin. Myelin is like a fatty lipid substance. It doesn't conduct electricity very well. And so the signal that's moving down your axon is really an electrical signal. It's a voltage, a change in charge. And so it's an electrical signal that's actually moving down the axon. And so what happens is that the axon only being exposed at the nodes, what advantage does that have? Well, when these sodium gates open and the sodium comes in and the inside of the cell becomes positive, that triggers the sodium gates down the axon, so it skips part of the axon to the next node where these sodium gates open, you get a huge influx and it becomes positive, which triggers this and so on. This makes the signal kind of, they say sometimes it quote unquote jumps. It literally doesn't jump, but it looks like that from node to node. And it makes it go faster that way, rather than if you had sodium gates all along and these guys open and these guys open and so on, it would be a much slower process. So this makes it happen much quicker and get to the end of your cell much faster than if it was unmyelinated. So that's the advantage of having myelin and increasing the speed. And so that's what this picture is showing you here. So the arrows are showing you the quote unquote jump, all right, and so they're showing you here the cross section. So at the nodes is where you have the sodium gates. If the sodium comes in, the inside of the cell becomes positive, which then skips all of this part here, and these sodium gates open over here, and so it triggers those to open, and that's the jumping process. That's called, it has a special name. This is called saltatory conduction. It's from the word saltaire, which means to jump. Let's put here the signal. I'm going to put quotes jumps because it's not literal. much quicker. All right, this next slide you don't have. I just put on here to remind myself to talk about it. So I want to talk about just some stats about an axon being myelinated versus unmyelinated, just to give you an idea as far as how big of a difference it makes. So the first bullet's kind of cut off, but it talks about how if you have an axon that's myelinated, an average speed in which the impulse travels down the axon is 120 meters per second. So here's a meter stick in your mind, times 120 meters in 1 second. That's how fast the signal travels.
That's microscopic.
So,
It's like,
yeah, so it's like, I mean, you can't even fathom like how fast that is. All right, so very, very quick. And I just wanted to compare that to an unmyelinated might only move a half of a meter per second. That's a huge difference, all right? A half a meter in a second versus 120 meters in 1 second. Only difference is having myelin versus not. So this Stimulatory conduction, a big deal, all right, in terms of speed. And then another thing I just added here, I found this, I thought this was kind of interesting. The advantage of myelination is also not only with speed, but also space efficiency. So we said that what could speed up the speed of the impulse is the diameter. So the bigger the diameter, the faster impulse, but also myelin. And so they studied like a myelinated axon like us, and the axon of a giant squid, which has a bigger, it says here about 40 times greater the diameter of like a vertebrate axon. And what they found is that the myelinated axon is actually faster than the 40 times greater diameter of the squid, and it's more efficient for us to have thinner axons and have myelination than having larger diameter axons, because if you think about space efficiency and how much space, I mean the axon's not this big, but but if you have a certain amount of space, you have a larger diameter, as it says here, that you can fit more than 2,000 of our skinny axons that have myelination in the same space as this giant squid axon, and so therefore we can transmit way more signals in that same amount of space, all right? And so it's more efficient for us than having these giant axons. All right, so then the last thing we need to do is talk about what happens at the end. So nerve impulses travel between the axon of one neuron and the dendrite of the next, or between an axon and a muscle cell, across what we call synapses, or the space is sometimes called a synaptic cleft. At electrical synapses, the electrical current flows from one neuron to another, and at chemical synapses, we have a chemical neurotransmitter that carries information across. And most synapses are chemical synapses, so I'm going to focus on that. And that's what we kind of talked about yesterday. So here I talked about the neurotransmitter released from another cell, binding to the receptors and so on. So we kind of talked about a little bit about that. We just need to talk about what happens for the other cell to release those neurotransmitters. So the presynaptic neuron synthesizes and packages the neurotransmitter in synaptic vesicles located in the synaptic terminal. This action potential causes the release of the neurotransmitter, and the neurotransmitter diffuses across the synaptic cleft and is received by the postsynaptic cell. So we have a picture here. Can I go too fast?
yeah.
Let's look at this picture and show what's going on here. So what we're looking at here is the presynaptic membrane. So this is the this will be like the synaptic terminal, so let's be like this part of the cell going to communicate with another cell. And so, and this is the postsynaptic membrane. So what part of the postsynaptic membrane is it? The dendrites. Remember, the dendrites receive the signal. And so, so that's what we're looking at. So let's look at step number 1. So the first thing that happens, notice here, here's your signal moving down your axon. So it's going down the axon. You get to the end. What happens at the end, the last part of your axon, the last node there, is your last sodium gate's open. So I'm going to draw here a sodium gate and show sodium coming in. And the inside of the cell becomes positive. That's depolarized. So now this is happening at the end. So what's going to happen is that depolarization, instead of causing the sodium gates at the next node to open, which there is no next node, what it does is trigger another gate to open. So here's step 2. Notice here, this says voltage-gated calcium channel. What does that mean? It's a channel that's going to allow the calcium ions through. And it's voltage gated, which means it opens and closes in response to a change in voltage. So guess what? At the end of your axon here, sodium just came in and your cell depolarized. That's what triggers the calcium gates to open. So I'm going to put here triggers this inside of the cell becoming positive, triggers the calcium gates to open. And so that's what we have here is the calcium gates opening as a result of that change in charge. So calcium goes into the cell. Calcium, notice here, has a positive charge. So when calcium goes into the cell is going to become even more positive, right? So the cell becomes more positive as a result of that. So then what does that cause to happen? So now look at here, the other guys here that we haven't talked about yet is these synaptic vesicles that contain these neurotransmitters inside. So if we look at step 3, all right, so step 2 is the calcium channels going open and calcium going in. Step 3, what's happening to the vesicles? What process is going on there? This is exocytosis, where this vesicle comes over here and fuses with the membrane, expelling its contents. And what is its contents? The contents are the neurotransmitters. So now we're at where we left off yesterday, or started yesterday, with neurotransmitters in the synaptic cleft. These neurotransmitters diffuse across and bind to these ligand-gated ion channels. Ligand gating meaning that they open and close in response to the ligand, which is the neurotransmitter binding to it. So these guys right here have a neurotransmitter bound to it. These gates are open. These guys don't. These guys are shut. And so remember, on the outside of the cell, we have a high concentration of sodium. So then that's going to trigger sodium to go into the cell. Cell becomes positive, which then, if it becomes positive enough, will trigger the sodium gates to open and this whole thing to happen, okay? And so that's how they communicate. Now this postsynaptic cell could be another neuron, like so the cell could be like, you know, this neuron releasing the neurotransmitters like I drew yesterday and communicating with another neuron and triggering an action potential. Or the postsynaptic cell, this is a motor neuron. Motor neurons can communicate with skeletal muscles. So sometimes the cell the neuron is communicating with is a muscle cell, and so a motor neuron leaves your central nervous system and goes to a skeletal muscle and triggers it to contract. So how does it trigger it to contract? It releases a neurotransmitter called acetylcholine, which binds to receptors on your muscles, which causes changes to happen inside your muscles, which then cause them to contract. So it doesn't have to be another neuron, and that's what happens at the end. Actually, before I do this, I want to talk about, let's go to that last page in your homework packet, because that had to do with this, and so I want to just correct that real quick before then we'll wrap up the notes. So it was this page here that we hadn't talked about. And so let's just label all these things. So it asks you to label the calcium channel. So this right here is the presynaptic cell, right? And this is your postsynaptic cell down here. And it doesn't ask you to label those, but that's what we're looking at. So this is the synaptic terminal. So this is representing the impulse traveling down the axon. So at the very end of the axon, you'd have sodium coming in and the inside of the cell becoming positive as a result of that. So that's where we're at. So what does that trigger to happen? So the first thing that happens is the calcium channels open. So where in the world are the calcium channels? All these little guys right here in this picture are calcium channels. So all of these and all of these are calcium channels. So the calcium channels open, and that means calcium can come in. So it asks you to label the calcium ions. That's these guys, all these little dots here that are not in the vesicles. All right, that's calcium coming in. So the calcium influx in causes the cell to become even more positive. So then it asks you to label the vesicles containing neurotransmitters. That's these guys right here. So all of these are your vesicles. Inside are the neurotransmitters. And so what are they going to do? As a result of the influx of calcium and the change in charge, it fuses with the membrane and expels your neurotransmitters. So now your neurotransmitters are out here. Where's the space that your neurotransmitters are? It asks you to label synaptic cleft. That is your synaptic cleft. These neurotransmitters are diffusing, and so they diffuse across the cleft, bind to what are these guys here? These guys are the ligand-gated sodium ion channels. Once the neurotransmitter binds to those, your sodium comes in, inside the cell becomes positive, and then you can trigger another action potential as a result of that. And so that's what we should have labeled on there. Okay, I know that the second link on here didn't work, and a lot of people then didn't do this little quiz here. So what I did is I copied it for you. All right. So one of the things that you picked up here today has 5 multiple choice questions on it. So some people figured out in the handout that I uploaded with the hyperlinks in it, it had those questions. If you scroll down to a next page, it had these questions on there, but most people didn't figure that out. So most people hadn't done these quiz questions, and so what I'd like you to do is just take a moment and try these 5 questions. let's go over the answers to these. So, number 1 says an action potential arriving at the presynaptic terminal causes. And so, so here's your action potential. This is the coming to the end of the cell, which is the terminal. So your last sodiums are rushing in, the inside the cell becomes positive. What that's going to trigger to happen is C. The voltage-gated calcium ion channels open, calcium rushes into the cell. So then, as a result of that, for number 2, what happens is A, the synaptic vessels are going to migrate to the plasma membrane. And I forgot to tell you, it says it releases acetylcholine. Acetylcholine is a type of neurotransmitter that's important to know to answer that question. So sorry, I forgot to tell you that. All right, so then it's releasing, we can just substitute in there neurotransmitters. All right, so then 3, acetylcholine, which is a type of neurotransmitter, has which effect on the postsynaptic neuron, and it should be B, the ligand-gated sodium channels open, sodium diffuses in. And then 4, if the postsynaptic membrane potential reaches threshold level, what does that mean? If the inside becomes positive enough, an action potential will be produced, and that is true, that's A. And then acetylcholine, or just neurotransmitters, is actively transported from the presynaptic membrane to the postsynaptic membrane. That is false. It's not active transport. It diffuses across, all right, to that. All right, so that is that. All right, so let's go back to the notes, and we're just going to wrap this chapter up. So, when that communication occurs, the response can either be what we call stimulatory or inhibitory. And I kind of use that example with the knee-jerk reflex that I'll talk about in just a second here. After release, the neurotransmitter can do one of 3 different things. Can diffuse out of the synaptic cleft. Sometimes it's taken back up by surrounding cells or reused or recycled. And it can be used again, and sometimes it's broken down by enzymes. And so, long story short, the neurotransmitter doesn't stay attached to the receptor on the neuron forever. And so, because imagine if the neurotransmitter that's released from another cell binds to these receptors and always stays bound, you're going to have this influx of sodium and you're going to have this impulse going all the time. And so therefore, what happens is the neurotransmitter binds for a brief period of time and can unbind, and one of these 3 things can happen to it. And so, because if it unbinds and it just stays there hanging out, it can rebind again, and you can have the same signal going on all the time. And so it can diffuse out of that synaptic cleft. So here it can diffuse out, all right? Or sometimes there are enzymes that will degrade it and break it down. So I use the example, it's in the next one. There's like acetylcholine as an example. There's an enzyme called Acetylcholinesterase that breaks that neurotransmitter down. Or sometimes the neurotransmitter can be taken back into the cell through endocytosis, the exact opposite of exocytosis, bringing it in and the neurotransmitter can be reused. But all 3 of those things cause the neurotransmitter not to be hanging out in the synaptic cleft anymore. All right, so that for this neuron to get another signal, it has to have another impulse here to release more neurotransmitters to then have to keep on communicating. Oh, and then the first bullet here, the response can either be stimulatory or inhibitory. I talked about this when we talked about the knee-jerk reflex. So you have this sensory neuron that takes information to your spinal cord, and then that sensory neuron communicated ultimately with 2 motor neurons, one that stimulated the quadriceps to contract, one that inhibited the hamstring muscle from contracting. So therefore there are inhibitory and stimulatory or excitatory neurons, and what makes them inhibitory or stimulatory is the type of neurotransmitter they're releasing. So that will inhibit or stimulate the receiving cell. So how do neurotransmitters work? Yes.
So you said that the neurotransmitters can be fused out of synaptic cleft. When they're just like out there, can they
Yeah, usually, but they usually eventually degrades itself, so, so it doesn't stay forever in there, so, but if you have just, it's so minute, the concentration, that even if it did get into another, just one of those is not going to cause an influx of enough ions to cause it. So the calcium, so this is just writing in bullets that picture we just wrote about. So the calcium gates open, synaptic vesicles release the neurotransmitter. The neurotransmitter binds with the postsynaptic receptors. The postsynaptic membrane is either excited or inhibited depending upon the type of neurotransmitter that's bound. And then the neurotransmitter eventually is degraded or recycled, and it stops communicating. And I gave an example here, a common neurotransmitter, acetylcholine. Although there's a typo on your sheet, the acetylcholinesterase. I have the A, and then I have cholinesterase. I don't know what happened to the CETYL. Do you see? You see that? So it's a typo. I don't know how that part of the word got deleted somehow. I don't know what happened there. All right. You see that on your sheet, acetylcholinesterase. It's missing this part here. So you want to. Gotta add that in there. I think it says acholinesterase. All right, so the last little bit is just about different types of neurotransmitters. So there's lots, our body makes, and our neurons make lots of different neurotransmitters. There's 5 different groups of them based upon their composition. So every neurotransmitter is composed of something different, has different shapes, and therefore binds with different receptors. But a single neurotransmitter can have different receptors that it can bind to, so therefore can affect different cells. All right, so epinephrine can change the heart rate and the breathing rate, as well as cause glycogen breakdown in your liver. All right, so that one thing. And so acetylcholine is the first example that we've already talked about. This says at neuromuscular junctions. That's when a neuron meets a muscle cell, and so it helps with muscle stimulation or contraction, memory formation, and learning. Then the biogenic amines. Amine means that they have an amino group. If you remember that from our functional groups. Examples we've talked about is epinephrine. We're going to look in just a second at an example with serotonin, which elevates mood when it's released. So it's derived from amino acid, hence the amine, it's amino group. And then amino acids, we're going to talk in just a second and give you an example with GABA. This is, notice here it says, an inhibitory neurotransmitter. So this inhibits certain neurons in the brain, and we're going to look at serotonin and GABA and see how they affect the brain and affect mood here in a second. Then there are Neuro Peptides, which are really short chains of amino acids. So it can be part of an amino acid, a whole amino acid, short chains of amino acids. Example, substance P and endorphins. These are our natural, they help with our perception of pain. And so they're kind of like natural painkillers that are released, like, endorphins are released during childbirth. And so in extreme pain situations, your body will make these to kind of help with the pain. And we have studied endorphins and have found opiates and made drugs, opiate drugs, because these chemicals, these opiates, mimic and are very similar to the shape, chemical structure of endorphins. They bind to the same receptors, and so therefore when we take in these painkillers, these opiate drugs, they bind to the same receptors and then help us with our perception of pain. And so therefore they're painkillers. And so I just added that in as an interesting side note. And then gases, nitric oxide and carbon monoxide are local regulators in peripheral nervous system. So that I just want to show you that some can be gases. They're not all like the liquid. And that's the end. And so with this, you have a sheet that looks like this. Let me explain what we have here. We're running out of time in this class here. Let me tell you the plan here. So this sheet right here that looks like this says exercise 5. Only takes about 10 minutes to do. And so you will probably get the first one close to being done. But what it does is give you examples of this excitatory inhibitory neurons that I've been talking about here, just to kind of give you an idea of what they're all about. And so that's what that is. And the second sheet here looks like this. My plan tomorrow, we're done with the chapter, so tomorrow 2 things are going to happen. We can review as well as I'm going to pass back your last test. All right, so those 2 things are happening tomorrow. So the plan is, while I'm passing back, part of your review is I made your own copy of my wonderful drawings. All right? And so what I did though is it's similar to the picture up on the board, but I put numbers kind of in the order of things occurring. And so part of your review tomorrow is you're going to look at this and see if underneath it you can write number 1 and write in words what's happening. All right, and so you're going to put like you're going to kind of make it a little essay with numbers, all right, as far as what's going on. And the same thing at the end when it reaches the synaptic terminal, what happens there. So that's part of your review. So that's what this is. My other 2 classes had a chance to do this all within the hour, so I won't give it to you for homework. If you want to do it right when you come in, then we'll go over the answers. All right, that's fine with me. All right, just an example of excitatory. All right. One thing with this is that the first letter, I guess I can just read it to you tomorrow, but well, we have time. The first letter in the first paragraph up here got cut off. So some people first hour had trouble figuring out what it was saying here. So I'm going to go ahead and read it to you right now. And so if you want to just write in the first letter so you have the directions. It's some neurotransmitters cause receiving cells to transmit signals, and other neurotransmitters inhibit the receiving cells' transmission of signals. Neuron X, diagrammed below, adds up all the excitatory and inhibitory impulses it receives. This particular neuron transmits its own impulses only when at least 2 neurons send it excitatory impulses at the same time. Each neuron sending it inhibitory impulses can cancel out the effect of one neuron sending the excitatory signals. Given the inputs outlined below, state whether neuron X would or would not transmit nerve impulses in each case. So what you're looking at here is one postsynaptic neuron, and you have 6. Or maybe it's 7, A through G, 7, neurons communicating with it. Some of those neurons are excitatory, it tells you along the top there, some are inhibitory. And so this gets to the point that one neuron can have multiple neurons communicating with it simultaneously. And so what this means, like if you look at number 1, it has neurons A through G along the top, and it tells you the Y or the N tells you whether or not that neuron that presynaptic neuron is communicating with that neuron at that time. So all the Y's mean yes and all the N's mean no. So in order for the postsynaptic neuron to reach threshold potential and therefore start an action potential, you have to have at least 2 excitatory neurons communicating with it. But the thing is that you have to have 2 excitatory neurons, but if you have any inhibitory neurons, it cancels out one excitatory neuron. All right, and so therefore you have to look at what neurons are communicating to it and figure out, is neuron X going to transmit a signal or not? All right, so that's that.
So basically, when you take yeses and no's, you have to have at least 2 yeses for it to be.
No, because you have to look. No, not necessarily, because
well, even if you just have 1 left, it's still.
Well, you have to look at the yeses. Are they excitatory or inhibitory? You have to look at those.
Yeah.
You can't just look at yeses and say, oh, there's more yeses.
So let's say there's 4 excites and like 3
Inhibitory.
inhibits,
And it's not going
then
to.
it's not gonna work.
That's right.
At least
Two.
two.
That's correct. You got it. You got it. And the second exercise gives you examples with your nervous system and your central nervous system with stimulants and depressants and the neurotransmitters serotonin and GABA and how they affect. But we'll